of a lot of different folks from a lot of different institutions. Some of the background information is coming, from folks beyond my group, but some of the names that you see here on the screen today have really supported me throughout my dissertation, and so I am indebted to everyone who has helped me so far. This is definitely a team effort. With that said, there we go. So a quick outline for today. I'm going to first introduce this interactive polling software just to kind of get a handle on who's on the call today. The funny thing about doing these kind of webinars is that, it's really hard for me as the presenter to get a sense of how everyone's feeling or who everyone is. So we'll use some of this interactive polling software to get started. I know that this webinar has been offered to a lot of students as a resource. So we're going to start by just going over some of the basics about emerging contaminants and organic contaminants to make sure that we're all operating with a similar vocabulary as we have this conversation today. I'm going to briefly introduce PFAS, a specific type of emerging contaminant that I study in particular. I'll also hopefully convince you that it's important that we talk about PFAS and other emerging contaminants in wildlife for a number of reasons. And last but not least, I'm going to show some results from studies across my field, as well as some of my own dissertation results, talking about emerging contaminants in Massachusetts Bay and the greater Gulf of Maine. So to be clear, today is going to be a lot of fun, I hope, but this is not a few things. First and foremost, this is not going to be a highly technical, comprehensive review of emerging contaminants in wildlife. I see some of my colleagues on here, and I'm so appreciative you're here, but if you want more technical detail about some of the stuff I'm sharing, I'd be happy to send some of my recent conference talks along to you, and we can chat further. This is going to be a high-level overview for a broad audience. I'll also be straightforward and say I don't really know anything about metals and inorganic pollution, so we're not going to talk about that today, but that is an important facet of understanding contaminants in wildlife. We're also going to really skim over some important classes of emerging contaminants like pharmaceuticals and novel pesticides and novel flame retardants. Each of these can be considered emerging contaminants, but they really deserve their own webinar in unto themselves. So we're just going to skim over them today. And I'd also like to point out that I'm looking at URI science today, not a reflection of some of the organisms. And finally, final caveat, I'm not a medical doctor. And so when we talk about contaminants, I often get some questions about, well, how is this contaminant impacting me? I'd be happy to point you to some resources. I have a Google document full of resources if anyone is interested in that, but I can't give you medical advice. I'm not really sure, what some of these contaminants mean for your specific health. So with that, I'm going to move forward. I would like everyone to please navigate to this web address. This is an interactive polling software where I will make a poll active, and that poll will then prompt you for a response. And if everything goes well here, the response will then populate in real time on my screen. So I'll give everyone a minute to get to that website, and then we'll move forward. Okay, so we're going to move forward from there. Now, at that website, if I figured this out properly, you should be prompted to put in a little detail about yourself. So I'd like to know, what's your current role? Are you a student? Are you an educator? Are you a scientist? Where are you coming from today? I like it. We already have a scientist. Awesome. UMass Amherst, great. Student. Engineer, great. Nonprofit. Scientist, I love it. So for your information, the The largest word in this word cloud is the most popular response. So it looks like we have students and scientists and a lot of folks in between. So welcome everybody. I'll give this just another minute to populate. I see some educators and volunteers on there. Awesome. Cool. All right. Well, great. Thank you so much for everyone joining. It looks like the poll is now full. I think I only get 30 responses, so I apologize if you didn't get your response registered here. But again, I'm really thankful that everyone tuned in today. This gives me a lot of good information about who I'm chatting with today. My next question is maybe a little bit about your background. I'd really like to know, how much do you know about marine pollution or emerging contaminants? Choose one of these options that most applies to you. This will help me understand whether I should kind of gloss over some things or whether I should, focus on particular aspects. Awesome. Okay. Awesome. So the poll is now full. Thank you everyone who responded. It looks like we have a fair amount of folks who have some knowledge and are interested in learning more, as well as a few experts. Welcome experts. I'm excited to chat with you all. And as I mentioned, this talk will really be focused on folks who are trying to learn or have a general knowledge in this and are still developing their background in marine pollution and emerging contaminants. So my last poll question for right now, I'd like you to respond, and especially for those folks who are coming from a background where they're trying to learn more about emerging contaminants, let me know what your biggest questions are. I can't promise to get to all of them, but if I know where your interests lie, I can try to make sure that within some of my slides, I touch upon your questions. Also to reiterate, it says it on the screen, but answers to all these polls are anonymous, so I can't see who says what or who is even responding to the poll. So please don't be shy. Awesome. Okay, I see some good responses coming in so far. I see effects on organisms, ways of detecting, how we can help, one's most concern. Citizen science groups, great. I don't know, valid response. Solution strategies. Can schools get involved? Excellent question. Okay, cool. We have some great prompts. I really appreciate everyone chiming in. Some of these I think are already baked into my slides. Others might be a little bit beyond the scope of this particular talk. If you do want to chat about some of these directly, I'd be happy to have some conversations about that offline as well if I don't get to your particular question. So with that, I'm going to go ahead and dive right in. So in general, as I mentioned, we're not going to talk about anything metal related or inorganic today. We're really going to focus on organic pollutants and organic emerging pollutants. And so we're going to start off by making sure we all share a common vocabulary here. And so organic in a chemistry sense has nothing to do with organic produce or organic commercial designations. As a chemist, when I say I'm talking about organic pollution, I'm talking about molecules that are primarily composed of carbon. These can be exceedingly diverse because carbon has a great capacity to bond to a lot of other atoms and functional groups. This is different from what I've already mentioned about inorganic and metals, which don't have that carbon backbone that defines organic compounds. And so we're not going to chat about that today, but be aware that metal pollution and inorganic pollution is definitely a thing. When we talk about organic pollutants, there are hundreds of thousands of organic chemicals in circulation today. And so some are produced naturally by primary producers like phytoplankton or plants. Others are purposely produced by humans. Some are indirectly produced by humans. But when we get down to it, we're really only concerned about organic chemicals that meet certain criteria. Basically, criteria that sets them up to accumulate in living organisms and or possibly hurt us. So as an environmental chemist, we define these criteria as PBT or PMT, with the P standing for persistent. Persistent means that a given chemical is resistant to degradation, so they can live for a long time in the environment. Depending on what organic chemical you're talking about, some can stick around in the environment for a few days or up to a year, while others can stick around in the environment for decades or even centuries. Another component of this criteria is that they're bioaccumulative. This means that these compounds can accumulate in living organisms. In order to be bioaccumulative, a lot of compounds that meet this criterion generally have to have a low solubility in water so that they want to partition into tissues or sorb to sediments, for example. Another element of this is toxicity. So we generally are looking for those chemicals which we know can cause negative health effects, either in humans or in wildlife. And the M part of this criterion is mobile. And the mobile element recently kind of got added to our understanding of what makes a chemical of concern as the EU Chemicals Agency suggested this addition, because we now know that accumulation or adverse effects related to organic pollution can be really system specific. So if a compound is really mobile and capable of traveling a long distance, it can reach a lot of different habitats, and there can be problematic to some of the most vulnerable biological receptors in those very remote habitats. If a given chemical meets these criteria, we say that it is a persistent organic pollutant, or POP. For students on this call, you're going to see that acronym used a lot throughout the presentation, so take note of that one. POPs are targeted and somewhat regulated by the Stockholm Convention. This was developed in 2001, and it's a global treaty that has the purpose of protecting both humans and wildlife from harmful chemicals in the global environment, and those chemicals that meet this PBT or PMT criteria. The overall aim is also to reduce or eliminate the production and use of these particular chemicals that meet these criteria. Move forward. So some examples of ones that we know a lot about. We have, oops, there we go. We've known about some forms of organic pollution for 70 or 80 years. Some of them are unwanted byproducts from industrial processes, so they're unintentionally produced. For example, dioxins and furans can be produced by industrial processes like chlorine bleaching of paper pulp, manufacturing of some pesticides or herbicides, or burning chlorine containing polymers like PVC. PAHs are another unwanted byproduct that we've known about for a long time. And this can be produced from combustion of really any organic material like fossil fuels, wood, or garbage, or tobacco. Whereas we've produced some organic chemicals directly. We use these for a variety of industrial and commercial applications. For example, polychlorinated biphenyls were once used as insulators and as part of pigments and coatings. They were banned in 1979. Polybrominated diphenyl ethers, or PBDEs, were used as flame retardants. These were regulated in the early 2000s, and we added these to a lot of textiles to ensure, or to try to ensure that they were less flammable. As well, maybe one of the more famous examples on the slides is DDT, a form of organochlorine pesticide, which was used a lot across, you know, the 50s, 60s, and 70s to control insects, but we found that this had some really negative impacts. So the molecules on the right, just as a side note here as well, they're listed in kind of the order on the slide. And students on here, I mentioned these acronyms again because when you hear discussion of organic pollution, you might not get the full long spelled out name of some of these chemicals. You'll often see these acronyms used in place. So because we have known about some of these organic pollutants for so long, we have a really good grasp on some of their negative effects. So for example, PCBs have been associated with reduced IQ in some children, in studies focusing on kids. DDT has been infamously implicated in the eggshell thinning in birds that really devastated bird populations across North America, across the 60s and 70s, and the list goes on and on and on. This is really not an exhaustive list. We know a lot about dangers related to some of these older legacy organic pollutants. But then we get to the point of our webinar today of emerging contaminants, or those contaminants that we really don't know that much about. I want to point out here that emerging contaminants are not necessarily new creations. Some emerging contaminants that I'm going to talk about today have actually been produced since the 1950s. The designation of emerging contaminant is more appropriately thought of as that we just don't know a lot about the extent of where they are in the environment or what they can do. And so on this slide, I kind of have a hierarchy of chemical uncertainty, if you will. And so this hierarchy contributes to what we call the expososome. And this was a term coined in 2005 to really describe the measure of all the given chemical exposures that an individual will experience across their lifetime. And so this starts before birth and lasts until death. And so within this totality of exposure, we are exposed to some chemicals that we know a lot about. This might include some of the legacy organic pollutants that I briefly just went over. And these known knowns comprise maybe less than 1% of the expososome. And this will vary a lot given an individual, you know, where they live and a lot of demographic variables. But in general, we know about maybe less than 1% of the chemicals that an individual is exposed to really well. And so when I say this, I'm talking about things like mercury or DDT, like I just mentioned. Beyond that, most of what we are exposed to over the course of our lifetimes are things that we don't know a lot about, which is really where this CEC or emerging contaminant designation comes in. So known unknowns, this constitutes maybe between 5 to 10% of the expososome, and these are poorly characterized or little understood compounds that we might know that they are there, but we really haven't taken the time to figure out how they affect us, or we maybe just recognizing that they're problematic, or these are chemicals that have been introduced over the past decade or two to replace stuff that we regulated earlier in our history. And then finally, we have this kind of larger outside ring of the unknown unknowns. And this is really fascinating to me because this constitutes maybe 90 to 95% of the total exposures that a given individual experiences. And so we don't know what those chemicals necessarily are, and we really don't know how they affect us. We don't know what we don't know, in other words. And so this emerging contaminant world is really rapidly evolving with a lot of analytical advances, but there's a lot of room to define and delineate what is in these two outer concentric circles. So with that, I'm going to move to one more poll. I'd like to hear if you have any examples of emerging contaminants that you already know about. For students in the webinar, I would particularly like to hear from you here. Pharmaceuticals, good one. Microplastics, excellent. Personal care products, awesome. PFAS, my fave, awesome. Great, I see a lot of excellent responses. Thanks everyone for filling that in. Definitely microplastics, pesticides, yep. Cool. Okay, great. If you have any final responses and want to add that in, it looks like we have some great examples of what constitutes an emerging contaminant. Moving forward from that, a lot of these examples are stuff that was just listed by this group. Examples of emerging contaminants include pharmaceuticals and personal care products, things like disinfectants that we add to our soap, some of our medication that we take, as well as some of the breakdown products of those personal care products and how they transform in the environment. We also have a number of endocrine disrupting compounds, or compounds that mess with how an organism produces or uses hormones. Disinfection byproducts are a big one. Nanomaterials, plasticizers like phthalates, novel flame retardants, novel pesticides. I saw in the previous slide that we had mention of microplastics and PFAS. We're going to talk more about those later in this presentation, but I want to point out here something that's pretty key to think about when we talk about emerging contaminants or organic emerging contaminants, that a lot of them that we're now dealing with today are hydrophilic or amphiphilic in nature. So at the beginning of this presentation, I showed you some examples of the legacy organic pollutants, and you may have noticed that a lot of them had ring structures, or what we would call aromatic rings behind them. That makes that contaminant really not like to be in water. We would call that a hydrophobic contaminant. And so we've learned a lot of lessons. We know that that ring structure and certain other molecular properties will make a compound not like to be in water and want to be in a living organism and accumulate there. If you remember back to our PBT criteria, we don't want to produce a lot of chemicals that accumulate in living organisms, because that can cause problems. So today, a lot of the contaminants that we now are investigating, they're not like some of the legacy organic pollutants that really hate being in water. We're looking into a lot of different iterations that don't mind being in water. We would call this hydrophilic, water loving, or amphiphilic. They're okay with being in water. So when we talk about emerging contaminants, this discussion also gets wrapped up with clean drinking water and clean surface water, because many emerging contaminants are potential stressors for water resources around the globe. We're not really going to get into that today. We don't quite have enough time, but if you'd like more resources about that, please reach out to me after the fact. I live and breathe this topic, so I'm very happy to share resources about what I know. Beyond this list, I also want to loop back to the idea of the expososome and unknowns. So this list and the word cloud prior to this, you know, we named a bunch of categories of stuff, but as I mentioned, we don't know what we don't know. So while we can kind of list chemical classes that we think are emerging contaminants, unless we take the time to measure the totality of chemicals in a given habitat, or maybe get information about the totality of chemicals that are currently in production, we really don't know what emerging contaminants are missing from this list. There's been a few studies over the past decades trying to better characterize how many emerging contaminants there actually are. Maybe by one study looked at the regulatory list from the US and Canada and identified at least 600. Other estimates have suggested hundreds, if not thousands, of emerging contaminants that we really just don't know that much about. So I want to point out here, maybe a central point of this presentation is not knowing their effects does not necessarily make emerging contaminants without risk. And so while there are plenty of organic chemicals that really don't do much to us, there are this subset that we do need to be concerned about. This lack of information about a given chemical does not equate to a lack of impact. And so as a kind of a roughshod example here, humans and wildlife aren't running around chugging glasses of chemicals and keeling over from acute exposure. If we're only looking for those kind of acute impacts, we're going to falsely delude ourselves into thinking that a lot of chemical substances are safe in the environment. Really, most chemicals impact us in sublethal ways over a prolonged period of time. And so these type of effects are really difficult to look for, particularly with emerging contaminants that we're just describing in terms of chemical structure or chemical properties. And so this gets to one of the challenges of wrestling with emerging contaminants. Just because we don't know exactly what it does not necessarily make it harmless. I'm going to switch gears right now and just introduce PFAS. I see some names on the call that definitely know what this is, but I also see some students on the call who might not have heard of this particular family of chemicals. And so if you're someone who has not heard of PFAS or PFAS yet, please take a screenshot of this slide. I encourage you to look into some of the amazing news coverage and stories about this particular family of emerging contaminant, because it's really the poster child for this idea of, just because we don't know about something, it doesn't mean it's harmless. We didn't know a lot about these compounds for a number of decades, and yet we're now finding that they're one of the riskiest emerging contaminants that we're dealing with today. They've been produced since the 1950s. They were actually invented as part of the Manhattan Project, but we really only learned about the wide range of their negative effects and their wide environmental distribution in the early 2000s. There have been some great news stories, and I think Karsten mentioned this at the beginning of the webinar, a recent movie called Dark Waters, documenting the story of how PFAS really gained a lot of regulatory and public attention. And in a nutshell, for the folks who may not be familiar here, some of the companies that produced these family of chemicals produced them and distributed them while not being transparent about the risks that they knew were associated with this family of chemicals. And some high profile court cases really blew the lid off of that in the early 2000s and up until about 2015. And so I encourage you to check out some of these narratives online that describe the history of PFAS and then some of the human health effects of them. We're not really going to talk about human health effects of PFAS today, although I know that's a big interest for a lot of people. If you're interested in that, I keep a running document of links. Please reach out to me after the webinar, and I'd be happy to share that with you so you can check it out. But now that I'm kind of done fangirling about this awesome story and some of the great media coverage, I'm going to move on to actually tell you what this group of chemicals is. So for students on the call, this is the only bit of chemistry I'll really show you today. These are just an example of what a PFAS structure looks like. So PFAS is actually an acronym that stands for per and polyfluoroalkyl substances. That's quite a mouthful. So we'll say PFAS. You'll also see it abbreviated as PFCs. Some groups call it PFA and then S. So the term covers up to about 5,000 different chemicals, but this number is increasing almost daily as we discover new types of PFAS. These are organic. If you kind of check out the two structures on the right of the screen here, you'll see that where the little junctures are, for those folks who don't stare at chemical structures too often, those little junctures are carbons. So this is an organic chemical. It has a carbon backbone. And they're also really defined by an abundance of carbon-fluorine bonds. The carbon-fluorine bond is one of the strongest bonds known in organic chemistry, so it makes these compounds extremely stable under natural conditions. An environmental sociologist who really tracks the history of PFAS has found evidence suggesting that some of the chemists that work to develop these compounds allegedly described their environmental persistence on geological timescales, meaning once they are in the environment, they do not break down. There are some PFAS structures that we would call precursors. These can break down, but they actually break down into other PFAS structures that may be more stable. For folks who are familiar, we have things like fluorotelomere alcohols that break down into the perfluoroalkyl acids, which are unbelievably stable. So beyond the stability, they also have some unique chemistry that's much different from those legacy organic pollutants I showed at the beginning of this. They can be in water just fine. They don't have that ring structure that makes them really hate water, in other words. They have a tail end that is kind of about water, and then they have a functional group that typically makes them okay with being in water. Their persistence and this kind of unique ability to hang out in water means that they can be, or excuse me, that means that they are ubiquitous in our environment. We put them in almost every imaginable consumer product, textiles, cosmetics, cookware, paper products, food packaging, outdoor clothing, countertop coatings, different paints, firefighting foams. We put them all over the place, and then they then leach out of those kind of different intended purposes and into the environment. Once they're in the environment, they can be transported by water, air, or kind of food web interactions. Now that I could talk about PFAS forever if you didn't just notice that, so I'm gonna try to contain myself here and move on a little bit. We care about these particular group of emerging contaminants because they have met a lot of that PBT or PMT criteria. They are extremely persistent, they accumulate in living organisms, and from what we know, they can be toxic at really low levels in humans. So for example, on the slide here, I put up a graph from a 2012 paper by Philippe Grandjean. He looked at children in the Faroe Islands from birth up until they were about 5 years old and tracked their immune response. And he did this by measuring antibody concentrations in those kids over time. He found that children in that population had a 50% reduction of antibody concentrations for each doubling of PFAS in their blood. I want to point out here that the amount of PFAS that were in those children's blood, that's an amount that you and I have in our blood right now in all likelihood. These are not environmentally very high levels. And so this was a huge finding because it shows that levels that we're exposed to right now in our environment, you know, we don't have to live next to a PFAS hotspot, and that has impacts in our daily life. Beyond this particular example, they are also associated with a number of different effects in humans as well as in animal studies, including impaired liver function or fatty liver disease, cancer at high exposure levels, different developmental disorders and reproductive issues, as well as issues with weight regulation and endocrine disruption. A lot of these studies have demonstrated effects in humans or lab-controlled studies. While we'll get back to this in a little bit, we're still figuring out what exactly these compounds are doing in wildlife. So these negative impacts are concerning, and maybe even more concerning when we consider just how many ways we're exposed to this particular group of emerging contaminant. As I mentioned, we add them to a lot of our products on a daily basis, and so that makes its way into our wider environment as well as directly exposing us through use of those products. This schematic was developed by Elsie Sunderland in a recent paper specifically for PFAS, but it generally applies to a lot of other emerging contaminants as well. And so for students on the call today, if you're kind of trying to picture, you know, how do we get exposed to emerging contaminants, definitely take another look at this schematic because it really outlines the key ways. You know, we get them from products that we use and we get them from our environment. and those environmental exposures include air and water and food as well. But it just so happens that we share our environment with millions of other living organisms who rely on these same natural systems that we do for food and water and diet. So we can leverage wildlife as a great way to better understand PFAS in human populations. This helps us get a grasp on the distribution and impacts of emerging contaminants like PFAS in the environment. And this also helps us understand how emerging contaminants actually impact us or could be impacting wildlife directly. Using wildlife as a tool to understand emerging contaminants is beneficial in a lot of ways. And as someone who actively studies PFAS, I would say first and foremost, it is much simpler to work with wildlife compared to human subjects. So humans do all kinds of crazy things, and there's a lot of ethical limitations on safely conducting research with humans. However, we harvest a lot of wildlife for food or encounter a lot of wildlife on a day-to-day basis. And so we can measure emerging contaminants like PFAS in those samples. And this helps us get around some of the limitations of measuring emerging contaminants in human, and also allows us to maybe more freely investigate what kind of emerging contaminants are where in the same environments that both wildlife and humans are relying on. As I alluded to there, we rely on the same natural systems as wildlife for our air and water and food. And so we actually can't tell the story of human exposure to emerging contaminants without looking at the living creatures and living systems that we rely on. So before we spend the time to conduct a study in human beings, for example, it's a great idea to look at emerging contaminants in wildlife and in the environment to see if they are meeting those criteria of persistence, bioaccumulation, and toxicity. So over the past few decades, the scientific community has really taken up the call to look for a variety of emerging contaminants in wildlife. Important advances in analytical instrumentation, and particularly liquid chromatography, mass spectrometry, have allowed us to rapidly expand our understanding of emerging contaminants in wildlife around the globe. We now know that many emerging contaminants can be found in every level of biological organization, from remote polar regions all the way to the bottom of the ocean to the upper reaches of the atmosphere, for example. On this slide, I've grabbed a few images of creatures that have been found to contain PFAS and or novel flame retardants, for example. You'll notice that we span a couple different animal kingdoms here and a lot of different habitats. And I hope this reiterates that emerging contaminants have traveled widely in our environment. Our expanding focus on emerging contaminants in wildlife has taught us a lot, though. For example, this study from 2017 looked at PFAS in whale muscle tissue from around the Faroe Islands. The Faroe Islands are demarcated in red in the globe in the top right. And so this study looked at a couple different kinds of PFAS, and they provided a lot of insight about how levels of two particular PFAS were changing over time. PFAS is a PFAS that we know is really stable in the environment and really likes to accumulate in living organisms. This particular study found that PFAS levels were not declining, even though we stopped really using PFAS in the early 2000s, in 2006 specifically. This same study found that another type of PFAS called fossa responded really rapidly in the whale tissue to regulation. So we said we weren't gonna make fossa or use fossa anymore, and that showed up very quickly in that whale tissue. This is really insightful because this tells us that when we make a regulatory action, it can show up very quickly in wildlife populations. This is another study that looked at multiple PFAS in seabirds from close to Antarctica from 2019. They found PFAS in all of the birds there. This means that PFAS are capable of making it to really remote regions of the globe that don't have direct human inputs of these chemicals. And they're making it there due to water and atmospheric transport, which are two pathways that we still have a lot to learn about for PFAS, as well as a number of other emerging contaminants. This study also suggested that species that spend the most time in the northern hemisphere have higher concentrations of PFAS. This is because the northern hemisphere is home to a larger proportion of the global population and many high-level PFAS sources. Whoops. So to bring the discussion closer to home, I'm going to loop back and start talking about some of my own research in Massachusetts Bay and the wider Gulf of Maine. And this area is an excellent place to study emerging contaminants for a couple big reasons. First and foremost, the area is ground zero for climate change in the ocean. It is one of the fastest warming regions of the global ocean. It's warming faster than 99% of the rest of the ocean. And so this is causing a lot of shifts in fish distribution, as well as invertebrate and plankton distributions that have cascading impacts in our food webs. In addition to that climate stress, there's an increasing abundance of floating plastics and marine debris in this area. And we also know that the land margins surrounding this area are becoming increasingly developed. And so there's increased waterborne transport of pollutants from developed coastal margins and a lot of coastal rivers that feed into this region. And this area is also atmospherically downstream of the US East Coast. So we receive a lot of atmospheric deposition of different pollutants from the wider US East Coast. So on top of all of these kind of factors or stressors, the area is also home to a number of important commercial fisheries and endangered megafauna like the North Atlantic right whale. In white here on the slide, you'll see an outline of Stellwagen Bank National Marine Sanctuary. It's one of 16 sanctuary and monument locations managed by the NOAA Office of National Marine Sanctuaries. And this area is one of the most productive regions throughout the Gulf of Maine, as a lot of upwelling and tidal mixing maintains really high levels of productivity here. And so a lot of fish and whales and birds take advantage of that and use this area as important feeding grounds or important provisioning grounds for their young. However, the Stellwagen is also a working sanctuary. So there's a lot of diverse stakeholders who also need to use that area to provide for themselves or derive economic benefit from the area. The culmination of circumstances surrounding this region makes it a great natural region to explore contaminants. In particular here, I'm really interested in how PFAS distribute in marine food webs. Some academic studies from a few years back indicated that seafood consumption is associated with higher levels of some of the larger or heavier molecular weight PFAS in American adults. So anything that has more than 8 carbons in its backbone, it's kind of a tracer of seafood consumption. This is really interesting and concerning to me because we know that those larger PFAS are more accumulative and they're associated with more toxic impacts. So I'm very interested and concerned to figure out how marine food webs in this area contribute PFAS in seafood as well as within the wider food web. As it turns out, it would be a huge lift to try to put together a study and narrow down exactly how marine food webs in this region distribute PFAS into humans around the Gulf of Maine, for example. But this is where seabirds and other wildlife come in because they present an opportunity to assess PFAS trends or other emerging contaminant trends across the habitats on which they rely. This idea of using seabirds or other keystone wildlife as canaries in a hypothetical coal mine has been validated across a lot of different scientific disciplines, as decades of research tell us that seabirds can be used as sentinels of the marine environment due to their generally predictable life histories and foraging strategies, their long lifespans, which include growth and reproductive events, their food web position as top predators, as well as their physiological interconnectivity to both air and water, meaning they rely on a lot of water resources, but they're also breathing air. All of this makes them sensitive to environmental stressors like emerging contaminants on observable timescales that we as scientists can measure. it just so happens that the Gulf of Maine is home to hundreds of thousands of great shearwaters. Great shearwaters are the species you see in the right corner of your screen right now. Hundreds of thousands of these birds come to the Gulf of Maine and Stellwagen Bank National Marine Sanctuary every summer. They're medium-sized seabirds that stay really far offshore. They actually can not take off very well from land at all. And they come to this region to gorge on forage fish and squid. The young individuals of this species also rely on the Massachusetts Bay area and Stellwagen area as a nursery area of sorts, as tracking data, which you see here in the presented GIF, tells us that young birds really book it to Massachusetts Bay after they learn how to fly so that they can fatten up for the first time on food resources in this area. So in collaboration with Stellwagen Bank National Marine Sanctuaries, I get samples from these birds to study contaminants and to study their diet. So I have a video of how that looks because it's actually super cool. We take a small inflatable boat and we go out to find rafts of these seabirds within Stellwagen or wider Massachusetts Bay. We net them with a long pole net and we bring them aboard. They're usually pretty upset about this, and they bite quite awfully. But this enables us to gather samples from live birds of blood or preen oil, which we can then use for contaminant analysis. Oops, no, go ahead. Okay, great. Beyond that, we also use samples from that get caught in fishing nets. As I mentioned, the overall region is a really productive area, and so a lot of folks are fishing out there. Some birds will get caught in those fishing nets, and these get collected for me and my team in collaboration with the Northeast Fisheries Observer Program. The bycatch birds come in, and I work with a team of researchers at Woods Hole to cut up these birds in kind of a standardized fashion to collect a number of different tissues and different samples for different folks to investigate. To date, we've cut up about 350 of these great shearwaters from the Massachusetts Bay and Gulf of Maine region, and this has provided us a lot of information about pollutants in the birds themselves, as well as diet habits of those birds that may be contributing to what pollutants they have in their bodies. So I'm going to share with you some results from one project related to the live bird and necropsy effort, where when I started my PhD, I set out to figure out, what do emerging contaminants look like in different populations of seabirds along the US East Coast. Because as I mentioned, while it might be hard to sample a bunch of human adult populations along the East Coast, we can measure seabirds to better figure out what sort of exposures of, or did I say that right? We can measure seabirds to better understand what emerging contaminants might be present in habitats that we rely on the East Coast for food and for recreation. So I had a number of individuals from Massachusetts Bay, those shearwaters that I just showed you about, as well as some herring gulls from Narragansett Bay and some samples from the Cape Fear River estuary. The Cape Fear River estuary has a really high potential for PFAS exposure that is downstream of an industrial PFAS production site, and some very high levels of PFAS have been measured in water there. Narragansett Bay doesn't have any PFAS production sources, but it's a lot closer to human sources of PFAS, like wastewater treatment effluent or human waste and landfills, and so that kind of has an intermediate potential for PFAS exposure. We found that all of the birds across all of the habitats had significant concentrations of PFAS in their livers. In this case is what this figure is showing. We found that those birds that were hatched near the production site had levels of PFAS in their liver that exceeded some of the known toxicity reference values that we have for PFAS. We also found that birds from Narragansett Bay and those offshore individuals, the great shearwaters, there was no real difference in the total amount of PFAS contained within those tissues. For some of the research folks on the webinar call, they had a mean of about 50 parts per billion in their liver of some PFAS. They did differ, however, in the types of PFAS. So the offshore birds were dominated by one type, specifically perfluoroalkyl carboxylic acids, while the inshore birds were not dominated by those. They were dominated by perfluoroalkyl sulfonic acids. Those are big, long chemical names just to say that they didn't have the same types of PFAS, even though they had the same amounts of PFAS. In this comparison study, we also identified a new type of PFAS. It has been previously identified in fish and water from the Cape Fear region. We found this new product in birds from both Narragansett Bay and Massachusetts Bay. And this novel PFAS is used in the production of a particular type of polymer and hasn't really been explored very much. This is one of those chemicals we would definitely define as an emerging contaminant, because we know it's there, but we really have no idea where it is in the environment and or what it's doing in living creatures once it gets into those living creatures. We also looked at a number of tissues from shearwaters as well as a couple other species for PFAS in an additional project, and we found that the birds contained significant concentrations of PFAS across at least 8 different tissues. And so while the hydrophobic organic pollutants that I showed you at the beginning of this webinar, they really like to be in fat. Yet when we looked at PFAS in all these different tissues, that's not what we see for this group of emerging chemicals. We see that they really like to be in liver, which is the dark blue on this slide, as well as in blood, which is the lighter blue. Just to orient folks here pretty quickly, I know it's a colorful slide with a lot of detail. On the x-axis, we have each different type of PFAS that we looked for. And on the y-axis, that tells you what percent of the total concentration in that tissue, or excuse me, that tells you how much of each tissue, each of those PFAS were found in. So again, we found most of it in liver and blood, which is much different from the legacy organic pollutants that we know a lot more about. I'm going to stop going on about PFAS here for a second to switch to another emerging contaminant. I saw this appear in our word cloud at the beginning of our session here to talk about microplastics. So the necropsy program that we have going allows us to look for stuff beyond PFAS. I might be particularly jazzed about PFAS, but we look for other things, including some of the legacy pollutants, as well as mercury, and now plastics. We honestly started looking for plastics because there were so many plastics in the birds that it was kind of impossible not to look for plastics in these individuals. We saved multiple tissues from each bird, including the stomach, which is where we look for those plastics. We then, oh, I thought I had a video. Guess not. We then look at those plastics by taking them out of the stomach. We specifically take them out of the ventriculus. The ventriculus is one component of a seabird stomach. It's a small muscular organ that kind of grinds up squid beaks or bones, or in this case, pieces of plastic. And it's not subject to regurgitation effects. So seabirds throw up a lot. If you work with seabirds, you know that they're pretty gross, and they'll poop on you or they'll vomit on you to try to get you to not bother them. And so by looking in this muscular part of their stomach, we kind of avoid that so that we're not getting a biased sample from the upper part of their stomach. Once we have them in a plate, we dry them out, and then we measure the length, the weight, and the thickness of each piece of plastic, as well as kind of characterize it by color and type. So far, this project has provided us a lot of interesting information. looked at over 1,000 plastic fragments from, I believe, close to 100 different shearwaters. We found that each bird in our sample set so far has ingested an average of 10 plastic fragments. These fragments are mostly between 2 and 10 millimeters in size, and this suggests that the birds probably aren't getting this plastic from their prey. They're probably eating those plastic fragments directly because the fragments are bigger than what most of their prey can accommodate in their digestive tract. Another big takeaway from our results so far is that 95% of the juvenile great shearwaters that we have looked at have ingested plastic. If I'm not mistaken, I think we have only found 4 birds that have not yet ingested plastic. This is a higher frequency of plastic ingestion compared to a study that was done in 2003 that looked at plastics in shearwaters. It was a smaller study, but they found somewhere between 60 to 70% of shearwaters had actually ingested plastic. This likely relates to the increasing abundance of floating plastics in the North Atlantic environment that supports these particular seabirds. This high rate of plastic ingestion is also sustained over the past 10 years, meaning in each year that we have samples for, a high proportion, 90 to 95% of birds have plastics associated with them. We also found that the juveniles had more plastics in them than the adults. Adults had a plastic ingestion frequency of about 60%. We also took all of those plastic fragments and bless my dear undergrad, Gwen. She worked with the Center for Coastal Studies here in Massachusetts and Christy there to look at the type of polymer that each one of those plastic fragments was. And so this was over 1,000 tiny pieces of plastic that were subjected to FTIR spectroscopy. This is a chemical technique that tells us what kind of plastic these little fragments are. If you recall back to that picture of all the plastics in the dish, it's really hard to tell what product that plastic came from unless you do some additional analysis to figure out what type of plastic it is. And on this particular figure, this is just to say the larger circles indicate a larger proportion of that particular type of plastic. Most fragments were polyethylene or polypropylene. This means that most fragments were also from recyclable plastics. It's pretty tough to ID what product each of these fragments initially came from, but the most common item ingested seems to be bottle caps. Bottle caps are usually made of polyethylene, and for some reason, we seem to find a lot of them in these seabird samples. They're really distinctive to pick out as opposed to some other types of fragments, because if you picture a bottle cap, the ridging along the sides of the bottle cap, that stays for a long time after the plastic has been in the environment. And so we see that ridging and some of the curves associated with those plastics, and it's indicative that it's a bottle cap. So I saw a lot of comments about, what are the effects of these emerging contaminants in wildlife? And we brushed over 2 big types of emerging contaminants today, PFAS and microplastics or plastics. And I want to highlight here again that emerging contaminants comprise a vast and very diverse set of substances and materials. So while we talked about 2 here today, there's a lot more to talk about with those 2, as well as kind of the wider world of emerging contaminants. If you are interested in any particular emerging contaminant that I didn't go into here today, please let me know. I'd be happy to chat more with you about it. I really live and breathe this stuff. So at the end of, I've given some iteration of this talk a few times, and at the end I'm always asked something like, well, how are these emerging contaminants impacting wildlife? And my answer to that is very clear. We really don't know completely. So we have some indication that, for example, PFAS will impact the lipid that we see or the fat that we see in a given seabird. We also have some evidence that suggests PFAS impact the reproductive ability of birds and other wildlife. We also have evidence that PFAS can impact the hormone production in marine mammals, for example. However, it's really difficult to identify impacts of emerging contaminants in wildlife, because in this photo, we see an acute impact. And so it's easy to identify a dead bird chock full of plastic as an impact of a contaminant, but it's a lot more difficult to pinpoint sublethal impacts from prolonged exposure to a particular type of PFAS. In addition, organisms aren't just exposed to one type of emerging contaminants. They are exposed to a veritable soup of contaminants, both emerging and legacy, that surround them on a daily basis. And while some of these are likely harmless and kind of just go about their merry way, some of these are definitely not. And overall, we need more information to pinpoint exactly what these are doing in our wildlife systems. And so I offer here that even though there are so many questions about emerging pollutants in wildlife, it's important to keep asking these questions about where emerging contaminants are and how they are impacting wildlife, because ultimately we as humans are tied to the health of these same natural systems that support wildlife. Exploring emerging contaminants like PFAS and plastics helps us understand these same pollutants in our own air and water and food, in our own bodies. And I believe Rachel Carson said it best when she stated in Silent Spring that we really need to stay tuned via scientific and cultural channels as to what contaminants we are allowing into our environments and what the risks are associated with those. Once they are out into the environment, it is much more difficult for researchers to go back and figure out their impacts versus finding that out beforehand and making decisions based on that precautionary principle. So with that, I have talked for long enough, I believe. Thank you so much for everyone who has stayed tuned. I want to point out I am part of a National Institute of Environmental Health Sciences Superfund research program. It's called STEEP. If you would like to take a screenshot here, the STEEP website has a lot of information specifically about PFAS, as well as some more information about my particular research in support of the center. I very quickly glossed over PFAS, but there are a lot of important questions related to this particular family of emerging contaminants. So I'd encourage you to definitely check out some of these resources further. I saw at the beginning of the presentation here a word cloud that asked, which emerging contaminants should we be most concerned about? I will admit my heavy bias here. I study PFAS and it's very important to me, but based on the chemical and biological evidence we currently have, PFAS are one of the most pressing environmental contamination issues that we are facing this generation. They are everywhere. We know they have impacts at really low levels in the areas that we have looked at so far. And so if you are interested in learning more about a particular contaminant, learn about PFAS. So thank you everyone so much for your time.
Great, thank you very much, Anna. So I'll open up the floor for questions. Susan or Birgit, have you been compiling some questions?
Yeah, there's a few questions already that we've received.
Okay.
So Barnett Ratner had asked about, you mentioned significant levels of PFAS in bird blood and liver. What is or are the toxicity references values for PFAS in birds?
Great question. So based on 2005 work for PFAS, one type of PFAS, we have liver toxicity reference values of about 140 parts per billion for PFAS in female birds. With uncertainty factors, I think there is a definition of toxicity reference values up to 600 parts per billion. And in the production sites birds, we had concentrations exceeding both of those values.
And then there's another question about, are the MPs particles an important source of emerging contaminants that get transferred into bird blood or liver?
That's a great question. So for a lot of emerging contaminants, they are not necessarily transported on plastics to the best of our knowledge right now. PFAS, like we mentioned here, they like to be in water. They're okay moving around in water. And so stuff like mercury or PCBs or DDT, those really like to sorb to things and get out of water. And so previous research has suggested that those legacy organic pollutants can be associated with plastic ingestion. But for some of the emerging contaminants, which like being in water, right now we don't think that they're associated with plastic ingestion. So my answer right now is no, we don't believe that plastic ingestion is associated with PFAS levels in the birds from my study.
Excellent. So somebody had asked whether where this recording will be posted, and I believe we'll get it onto our CSCR website as soon as we have an opportunity to do so. So our website is ccscr.org. That stands for an extra C for Cohasset, Center for Student Coastal Research.org. And it's also going to be on 143 TV, which is our local television station here in Cohasset. So I'm Susan Bryant from the Center for Student Coastal Research, and I'm just handling the technical parts of this Zoom. So I just wanted to remind people that if you can find the chat, it should be sort of in the middle of your toolbar in the bottom of your Zoom screen. And you can hit chat and type in to everyone a question that we will pass on to Anna. If you are unable to do that, there's a little thing that enables you to raise your hand with a little fake yellow hand. But if you want to just raise your regular hand, I think I can set my screen so I can see that, and then we can unmute you and have you speak in the regular non-robotic way if that's easier for you. So type fast your questions, or raise your hand on your screen, or raise your little virtual hand that comes in next to, where is it, reactions? I forget. Okay, question for you, Anna. Do PFASs or PFASs concentrate in the Arctic due to water and air currents?
That's a great question. And so I'm not sure if concentrate would be the right word there. We do know that they definitely make it up there related to long range atmospheric and waterborne transport. I think to the best of my knowledge, different PFAS are transported through different pathways, along different ways. So carboxylic acids are more waterborne versus the PFAS and the sulfonic acids. They are likely to be transported via precursors through the atmosphere that then get transformed or oxidized in situ or in vivo into PFAS and those sulfonic acid PFAS. So in terms of which one is more important, air versus water, I'm not exactly sure. If whoever offered that particular question, I'm picturing a couple papers that did some great modeling work in, I think it was 2012 and 2013. I'd be happy to send them your way if you want to check into it a little bit more. Thanks for the question, by the way. That was a great question.
Any other questions, students, CSCR students, other high school students? Any high school students out there with questions? So our audience at CSCR often includes our students who participate in our research programs. And how about any college students with questions? All right, going up the team.
Hold on, while we're possibly waiting for some questions, I actually have one because I think I do recall, Anna, that PFAS can be already detected in raindrops.
Yeah, definitely.
And obviously, since many water utilities have groundwater resources, it gets washed into the groundwater and ends up in our tap water. Are you aware that water utility companies are actually monitoring now all PFAS? Because I wouldn't even know whether, for instance, our town is doing that.
Yeah,
and I do actually recall that I think one town in Massachusetts, I don't recall the name, had problems with PFAS. And for some reason, I Googled it, and you know, since social media is ubiquitous and picking up my Google search strings or whatever it is, I now get, for instance, advertisements for PFAS filtering solutions. You know, what are your comments, or what do you know about this?
Yeah, so PFAS are a sensitive topic for water resources in that water utilities don't put the PFAS there. Consumers and industrial producers of PFAS put the PFAS into the environment. Karsten, as you mentioned, PFAS can be rained out. More likely, they're transported through groundwater. They like to be in water. So once they are in the groundwater, they are then in that water resource. We have a number of identified ways to get PFAS out of groundwater. All of them are expensive. One of the primary ways is using carbon to filter PFAS out of drinking water. This is currently being done in Barnstable, Massachusetts. There are a number of resources on the STEEP website that I shared, and that is an active research site for my particular program, looking at PFAS in that groundwater source. And so PFAS are not regulated formally in the United States. They're regulated at state levels in general. The federal government offers health guidelines, whereas a lot of states have set lower limits for their drinking water resources, which then require water utilities to look for PFAS at those levels. The state-specific levels are across the board and different from one another. But depending on what provisions are in place in a given state, there's no federal requirement to look for these in drinking water. So that is a rapidly evolving area of regulation and in research. If anyone has particular questions about that, I have a document about drinking water and PFAS. That is not something I actively study. I study surface water and wildlife, but I can point you in the right direction if folks want to reach out for that.
Yeah, I mean, we should probably definitely share that, and we can try maybe put it on our website. What do you think, Susan?
Yes, we can try to do that.
Cool.
Okay, so are there more questions? Because I think we wanted to keep this event at 1 hour. We're a little bit over it already for 15 plus minutes. How are things looking? Are still more questions coming in, Susan?
I don't see any other questions on the chat. I don't think I've missed any.
Excellent. So I'm putting my my email address into the chat. For some of the students who I know were on today's call, please, if you have questions and maybe we're a little shy about it, please just email me directly. I would love to chat with you. I was so excited about this particular opportunity to present, knowing that there was going to be students involved. So please don't hesitate. If you're interested in emerging contaminants, or if you have questions about something I didn't talk about, please shoot me an email.
Wonderful. Thank you so much, Anna. That was awesome. I think we all learned a great deal more. We are concerned a little bit more, you know, not that we're having too few things to be concerned about these days, you know, that's why we're doing this online lecture. But thank you all for attending this webinar presentation. And again, Anna, I want to thank you. Everybody take a silent clap here. Most of us are muted, so you won't hear it. So we will make the presentation slides and the webinar file available. We'll try to do that here at the CSCR on our website. Again, if you have any questions, contact Anna directly. I know that she also replies on LinkedIn. That worked well for us. Thanks, Anna. Also, contact the CSCR team directly if you want to know more about what we do. And Susan has given you the website previously. Also, check out our website for updates, future events, also webinar announcements. So, Susan, I'll leave it for you to make any final announcements. And so I'll end the official part, and feel free to log out and go wherever you have to go, and we'll wrap it up here from our end.
Okay.
Thank you again, Anna. Thanks so much. That was
Bye,
awesome.
everyone. Thank you.
Thank you, Karsten. So there's one other question that looks pretty interesting. I'm just going to add it in there if you don't mind. Indigenous communities that might consume fish blubber, have they been studied for high levels? Do you want to take that now or get back to that person? I'm curious.
Yeah, sure. I can try. So I think that this is an area of really active research, actually. I know that there's been work in Arctic indigenous communities finding really high levels of PFAS as well as legacy pollution, but also, oh gosh, if the person who offered that question reaches out, I'll send the study. I don't have it memorized, but I do know that this is a particular topic of importance for indigenous communities who rely on wildlife for food resources. And yes, they typically they typically display higher concentrations of a lot of different types of pollutants.
Great. So I have a question that will be a segue into our next part. I always like to ask our presenters, when you think back on where you were as a middle school student or as a high school student, was there a certain interest you had or a certain experience that you had that sort of propelled you into the direction that you are now?
Oh, that's an interesting question. I've always been really interested in science. I'm pretty curious. So I've cycled through a couple different scientific endeavors. I don't think I realized I wanted to be an environmental chemist until I figured out later in my probably high school or early college. I grew up in the backyard of an area that has really high levels of emerging contaminants. And that's also surrounded, it's a pretty poor community. And so there's that really, it never sat well with me that we never really knew what contaminants were in our environment. And so I've become quite an ardent, advocate for figuring out what contaminants are in the environment around you, because everyone really deserves to know that their air and water is safe and healthy for them.
So that sounds like the takeaway from that little statement is if you have something that doesn't sit well with you, or if you have something that you're curious about, you can go forth in the world and learn about it and figure it out.
Yeah,
so that's really exciting.
Absolutely, and you can make it your job, and it'll be so much fun.
Yeah, chasing after the seabirds looks pretty fun, I have to say.
Yeah, totally.
So I just wanted to say a couple of things about what the Center for Student Coastal Research does here in Cohasset. We are just launching into the high season for our summer research. So we are doing field expeditions. I was out in the scenery that's behind me, my little fake background there today with students who are researching eelgrass. And we had students in the lab from 7 in the morning, hi Grace, to 3:30, no later than that, in the afternoon. And they were working, the ones in the afternoon were working with Doc T, who is a very much beloved chemistry teacher. And she was describing all the different things about how things work in the world. And I was fascinated, just sort of listening in on the side as she was talking to the students. So we run a bunch of project-based learning opportunities for students. At this point, they've been middle school and high school students. And, but as education is in a bit of a strange place right now with the social distancing and a lot more online learning, we've often thought about expanding toward college students, and it's kind of an interesting time for seeing where we can go. We do happen to have some CSCR alumni on this call, and I will, let's see if I can, I'll unmute you guys as far as I know, and I will let you take it from there. Or maybe you can unmute yourselves. Okay, yeah. So they have been discussing a little bit of what they've been doing since CSCR, and they come from a different levels on the number of years since leaving here. But as we say to our students, you can check out anytime you like, but you can never leave. I'm stealing Jack Buckley's thunder here. And that really means that we really love to hear about our students and where they're going. We love it when they come back. We're happy to have them come into the lab and say hi to people. And we feel like we're part of their network and they're part of ours. So let me have them introduce themselves. unless you'd like to do that, Jack Buckley. And alumni can take it from there. Can I mute you?
Does anyone want to go first? I can go first. Are we gonna, Jack, did you have a plan for this, for introducing us? Do we want to do an intro of what we're doing first?
No, I didn't necessarily have a plan because I didn't know who would be here. So for those who are still on, I think I see everybody in my full screen there. So for those of you who are still on here, we've been talking recently. Oh, I can do my own. There I go. Undo that. There we go. All right. So, Anna, by the way, that was an awesome lecture. So thanks so much. It was wonderful.
Thank you.
So thanks for sticking around a little bit too. So we've been reaching out to some of the students who are part of our alumni group and have made contact recently with Maggie, who just started to speak, and Tucker and Christine and Levente, and there were a couple other people on here earlier who I think have left. But anyways, we wanted to build up a network and try to get more of our alums to get together and try to create an opportunity for current students to connect with alumni and alumni to connect with people like you, Anna, and other researchers. And it's very much a first shot at it. It's very organic. We don't know exactly where it's going to go. And that's a good time for me to say, let me turn it over to Maggie, who can tell us a few words about what she envisions with this network and who she is and what her pathway's been.
Thanks. So yeah, I think I can talk more about who I am and what my pathway has been. I think Jack's vision for the network is a little bit more clear than mine, but basically my name is Maggie O'Grady. I was a graduate of Cohasset High School in 2012. Feels like forever ago. Oh my God, that was 8 years ago. I just realized that it's 2020. And I participated in CSCR all throughout high school, you know, going through during the summers and doing the water quality testing and looking at vegetation in the marshes and all of that fun stuff over the summer. And then my senior year of high school, I won a grant to do research on the sea foam in the Gulf River, which led me to going to the Massachusetts State Science Fair at MIT, which was kind of the highlight of my high school experience. And that was obviously an incredibly valuable experience and led me to, I mean, I was always really interested in science and knew that that's what I wanted to do throughout school and for my career, but definitely participating in the science fair and participating in CSCR just drove me to do it even further. And I ended up going to MIT for my undergrad, and I have a bachelor's in biological engineering from MIT. So I graduated college 4 years ago, and that led me to the Peace Corps in some convoluted way that doesn't really make sense based on anything else that I just said. But I've always been really interested in public service and volunteering, and you know, the Peace Corps was just kind of a natural next step because of that. And now I am working as a program manager at a nonprofit research organization in Boston. So that's kind of my life story. Who wants to take it from here? Did I miss anything?
I don't think so. I don't think you missed a thing.
Thanks. Christine, you unmuted.
Sure. So my name is Christine Guiney, and I'm a 2015 grad of Cohasset High School. And I started at CSCR when I was in, I think, the 4th grade, maybe, pretty young, doing some junior marsh monitors. And then I did a lot of work with the Gulf River with Doc T, and ended up doing my senior year an independent study, putting together a proposal, or just kind of putting together all the research we had found on the effects, the chemical and biological research we collected over the many years at CSCR to figure out some of the herring issues on the Gulf River, which was really cool. And I think then I went on to UMass Amherst, and I got a degree in civil engineering. And I think CSCR was definitely a foot in the path of going to civil engineering, because it includes environmental engineering. And I've always been interested in architecture and buildings, but also in marine biology and everything that CSCR was. I always enjoyed my summers there. And I just graduated in 2019 from UMass, and I worked for a couple months with a civil engineering company doing work in their water wastewater group. So a lot of sewer pipes and water distribution pipes in towns, because in Massachusetts it's pretty old, so they're doing a lot of quality assurance on the pipes in these towns to make sure water is being flowed distributed to all the houses and stuff. And yeah, right now I'm just figuring out my next couple steps, but that's about it.
Nice. Cool. Should I go?
Might as well.
Cool. Awesome. I'm Tucker. I was in Maggie's class actually at CHS. We graduated together. And yeah, so I feel like for me, CSCR taught me how to connect the science, how relevant science was, in the broader communities, and sort of instilled in me, then understanding the science and bridging that into society is a very tangible and important thing to do. And I learned that through things like, the Phragmites study that I was working with, took a look at, sediment bacteria in the harbor and related it to, water flow, and was involved with a variety of projects as well early on. I don't remember all of the specific ones I was part of, but I just have a lot of memories of being out in the marsh and, collecting the water samples and, measuring the salinity. A lot of good times, for sure. So since then, I'm an engineer. I got a degree in mechanical engineering and was in school focused a lot on how engineering could be used in social entrepreneurship in sort of the international development context. Since then, I've moved into medical device development, so product development for medical devices, some consumer products here and there, but sort of connecting back to what I said initially, I think CSCR has impacted me to this day because medical devices, in my mind, is a really great way to connect the science into a direct impact to people. So that's the quick bio about me.
That's awesome. Well, it's great to see you guys again. And I don't know, so it's just a few of us here. Birgit is a new member of our board, and